Huy Nguyen from Iowa State University. He's going to talk about tracing the ancestry of operons in bacteria.
Thank you. Hi, my name is Huy Nguyen. Yeah, I'm from Iowa State University in Computer Science program, and I'm working in Professor Ido Freeberg lab. And in our lab, we are interested in the evolution of complex system. And today, I will talk about a model I have developed to look at the evolution of gene block and operon in bacteria. So the motivation is to understand the evolution of complex system. And the question here is, how do gene block evolve in bacteria? Then we will introduce our approach with use of maximum parsimony to find the most optimal gene block. And then I'll discuss some of the results and future work. So what are gene blocks? Gene blocks are genes that are co-located and usually sharing the same pathway or protein complex. And an operon is a special case of gene block where the genes are co-transcribed by polycistronic mRNA and under common regulation. So how do we describe the evolution of gene block? Previously in our lab, we have discovered there are three main events. So let's say that we have a parent gene block of gene 1, 2, 3, and 4. And the bottom part are the possible children that could have evolved from their parents. We define there's a split event if between gene 2 and gene 3 in the children genome have a distance of more than 500 nucleotide away from each other. And that is a split event. And we say there's deletion event if we cannot find gene 4 in the children gene block. And there's a duplication if gene 4 is duplicated right here. So here's our main problem. Ancestral reconstruction problem can be stated as follows. So giving a reference operon or gene block in your reference genome and a phylogenetic tree where each leaf node are populated by an orthologous gene block to that reference operon. The question here is, what are the ancestral state at node 6, 7, 8, and 9? So our approach basically using maximum parsimony, which using the least amount of assumptions. Here we also assume that gene loss is more conceivable, probable than gene gain. And here we present a toy example, which is basically dealing only with the deletion event. And so as you can see, right here, we're going to traverse in post order from leaf node to root node. So at node 6 right here, you have two candidates for the gene block, which is ABCD from node 1 and ABC from node 2. So I choose to include ABCD for the following reason. As you can see, gene ABC appear in the 2 of the children. So you're most likely to have it. I also include gene D because that gene D appear in 1/2, which is half, 50%. So that's why I also include it in the ancestral gene block. Then at node 7, now you have a candidate of ABCD gene block from node 6 and AB from node 7. Here I choose to include ABC only for the following reason. Gene AB again appear in 3 out of 3. And then gene C appear in 2 out of 3, which is still greater than 50%. However, gene D now only appear in 1 out of 3, so that's less than 58%. So it remove it. So using the same idea, I go through for the remaining in the node to the root node, and here's the final result. So that is basically the idea of our local approach with only here only demonstrate for the deletion event. So we have applied our method on several operons from E. coli and B. subtilis. Today I will show you a result on reconstructing ancestral on gibberellin operon. Gibberellin is are hormones in plant growth and development. They are known to be produced by some phytopathogenic fungi, and recently we also found that they are produced by some symbiotic rhizobia. So down here is the pathway for the operon in the symbiotic rhizobia. Most of them contain gene CYP15, which produce the compound of GA9, which is not bioactive. Only a few of them which contain gene CYP115, which can transform GA9 to GA4, which is a bioactive form. So from here on, I would denote a star to say that it is basically bioactive. So we apply the reconstruction method onto the gibberellin operon. Again, you have the reference species, which is Scythomonas oryza, and which have the reference operon right here. you have the phylogenetic tree with the, each of the leaf node populated by the orthologous gene block to that reference operon. So using the reconstruction, you can see that most likely in the beginning of time, the species will have all the gibberellin operon. So therefore, we put a star here indicate that it might have the GA4 bioactive form. However, throughout the evolution, it might have lost the gene CYP115, which is denoted by letter A. You can see that there's no A in those. Then it regained in two separate clay, which is in Cynorhizobium and Mesorhizobium. So we hypothesize that in those two, we might have that GA4 bioactive form again. And then one other interesting things is that in those that don't have the CYP115, they have fragment of it, which is basically like pseudogene of it. Then we also found that the pseudogene of CYP115, which is denoted by letter P, regain into the gene block at those two clay. We don't have an explanation for it, but it's a quite interesting observation. And lastly is you can see that in those two group of species, we have the full GA operon. Although they are quite distant in terms of species, but they have the full and in the same order as well. When the species between them don't have the full operon. So we highly doubt there would be a horizontal gene transfer that happened right here. So in conclusion, we have developed a method to reconstruct the ancestral gene block using maximum parsimony. We also provide some good visualization of the evolution process. In the next few versions, we would like to account for horizontal gene transfer and gene order. So, okay, so that just happened. So I would like to thank Professor Ido Freibert, Professor Oliver Ornstein, Ashish for helping me with the manuscript. I would like to thank NSF and Iowa State University for funding me, and for my lab for giving me constructive advice. I would have to QR code for my paper and for the code I have, and well, it doesn't show, but please come by my poster later this evening, SB 805. So that's it. Thank you.
Yeah, we can have a couple of questions.
I have a question about how your method deals with situations where you have incomplete data, because we clearly don't have like a full picture of all bacterial strains that exist. There might be families or genuses that are missing. Would this approach be limited in those situations?
That's a very good question. So for the data that we have right here, in our paper, basically we deal only with full, complete genome. But here, as you can see, most of the species right here might not have the full genome. So the way we do it, basically, we wrote some automatic script to go and pull all the context data, then combine them, then annotate them using broker to, in a sense, having a full, full pseudo of the genome to the best like capability that we can have. And then we can still use this approach for it. Yeah, does that answer your question? Okay, cool.
Yeah, any other questions?
Yes.
So you say you are finding a most parsimonious history.
Yes.
So are you minimizing duplication and losses? Or
Oh yeah.
Costs for losses?
Yep, I do. So for the local, it guarantees to have minimal because for the local approach that I showed earlier, it will guarantee the local minimal of the deletion and duplication costs for any parents and children node. For the global, it also guarantees for the global duplication deletion. However, for the split, it doesn't guarantee, but it's within the bound for it. So my answer is not exact, but it runs in polynomial time. So yeah.
Have you checked the effect of the tree? If you perturb the topology, do you get less parsimony?
Yep, that is one thing that we did. Like we, instead of building phylogenetic tree, we built some gene tree that using some gene in the operon. It would demonstrate like lesser amount of event because it tell you a more closer look at the evolution of the gene block. So yeah, we did that too. Yeah.
Okay, yeah. Let's thank for And yeah, our next speaker is Ana Maria Rosas.